Hello, my name's Mike Kennedy. I'm a assistant professor in environmental science at the University of Coventry in the UK. I'm also associated with the research centre in the university called the Centre for Agroecology, Water and Resilience. I've been asked to talk today about some of my relatively recent research work in Southern Africa, developing biomonitoring methodologies, primarily using macrophytes, that being my main area of interest and expertise, but also working with colleagues from other universities, the University of Glasgow, University of Zambia, Potchefstroom University in South Africa, and other South African colleagues at various institutes. We had a program called SAFRAS, Southern African River Assessment System, which was developing methodologies for monitoring water quality using macroinvertebrates, using diatoms, and also using macrophytes. I was leading on the macrophyte monitoring of various water bodies around Zambia. This was ongoing for a period of about 7 or 8 years, so we were building up data sets. The primary aim of the project was to build up those baseline data sets, but also to associate occurrence, abundance, species presence to various water quality parameters. These included things such as ground alkalinity, electrical conductivity, so things which could be related to groundwater conditions. So my background is primarily in plant biology and ecology, freshwater ecology, but our program was looking at things which could be associated with geology and of potential interest to geologists. So we did a lot of sampling across Zambia as a whole, lots of spatial sampling, lots of temporal sampling across several years, and collecting data on macrophytes. It was a successful project. We got a lot of data. We increased the records for IUCN red data book species, such as a species called Nymphea divaricata, which we found was associated with headwater streams, associated with the Zambezi Basin, incredibly low conductivities. So things which would equate to distilled water or battery water, and they weren't found in any waters that were higher in terms of alkalinity. So they've got a very restricted distribution, so we know we need to protect those waters, protect those headwaters in order to conserve rare species. Zambia as a whole is divided into 2 main basins. We have the Bangweulu Mweru headwaters, which ultimately flow into the Congo basin and towards the west of the country, and we have other waters. Most of the other rivers flow into the Zambezi Basin, which ultimately flows to the other coast of Africa. We did see some differences in terms of the Bangweulu Mweru headwaters that flow into the Congo. Very different plant communities associated with very low levels of alkalinity, very low electrical conductivity, but also at high altitudes. So again, this is to do with some of the various rock formations, laterite soils, etc. within those areas. So again, very low conductivity waters, but also an area of the country that is underdeveloped, but is under potential pressure from human population expansions, establishment of new towns, villages, and farming blocks. So activities which might impact and affect these kind of waters. And obviously the waters will have very low capacity to absorb these potential inputs of nutrient. On the other hand, we had areas, plant communities within the Zambezi Basin, which are associated with very high levels of conductivity and electrical conductivity up to about 6,000 microequivalents in areas. So very, very rich waters, far richer than we would get in the UK, for example. And we saw a differentiation in plant communities in relation to this. Quite often the plant communities were more species poor. This work was backed up by groundwater sampling And we could see in many cases there was potentially a movement of groundwater which supported many of the surface water bodies. So again, we use ground alkalinity, conductivity as indicators of the residence time of the waters within the geology, but also using conservative tracers such as oxygen isotopes and deuterium, which also indicated that there was an input of groundwaters into many freshwater systems. So that's just a very quick overview of what the project was about. So we were also looking at macroinvertebrates and diatoms as other biological proxies for monitoring water quality. But we can see that there is the potential to look at groundwater inputs from macrophyte communities present. We still consider it to be an area of ongoing research and still in discussion and planning with the University of Zambia to try and continue this monitoring via PhD students or other areas of activity where we can. I hope that's given you a brief idea of the research that we do and some of the applications it might have. Okay, thank you.